Hello guys, welcome to my YouTube channel. So today we are going to discuss Agrobacterium tumifaciens mediated plant transformation. So there are two components. One is the Agrobacterium itself. Okay, here we will talk about Agrobacterium, and more specifically it is Agrobacterium tumifaciens. Okay, and this is the pathogens that we will talk about, and plant is the host cell. So this is the plant cell is the host cell, and Agrobacterium tumifaciens is the pathogen which will help in the transformation. Okay, so what does this pathogen do? In general, this pathogen infects the host. So this is the basic mechanism. So what happens when a plant cell is wounded? So what happens when the plant cell is wounded? It releases certain chemicals called as the sugar and the phenolics. Okay, so sugar and phenolics. So these compounds are released from the wounded portion. Now these two compounds act as the inducer. Okay, so from these two compounds which are released from the plant cell, these are released from the wounded plant cell. So these compounds act as the inducer, and they induce a series of downstream events. So first it activates a protein which is called as the VIRA. Now first, before coming to the mechanism of this transformation, you have to know that Agrobacterium tumifaciens, this bacterial cell, contains as a general, this is a plasmid. Okay, so it has a plasmid. Okay, and in this plasmid there is one virulence region. So this portion is the virulence region, and this is the tDNA region. So this TI plasmid, so this plasmid is called TI plasmid. So TI plasmid is made up of broadly two components. One is the virulence region and another is the tDNA region. Now this virulence region on translation produces certain proteins which are called as VIRA, VIRG, VIRD1, D2, VIRV, VIR E2, and sometimes another protein which is VIRF. So this protein has been reported in some studies. So broadly, these 6 kinds of proteins, or 4-5 kinds of proteins, are translated from this virulence region or VIR region of TI plasmid. And tDNA is actually that part of the DNA which is actually transferred to the plant cell. Okay, so this actually is the transferred to the plant cell, only the tDNA portion. Now what happens when sugar and phenolic compounds initiates VIRA? VIRA activates its downstream protein VIRG by phosphorylation. Okay, so this VIRG protein is activated by phosphorylation, and what happens? It acts as a sort of auto-inducer. It acts on the VIR region and helps in the synthesis of other downstream proteins as well as its own protein. So this VIR D1, D2, VIRB, VIR E2, these proteins all get translated from this VIR region. Okay, now what happens? VIR D1, D2, this complex comes and just cuts the tDNA portion from the TI plasmid. Okay, so just this region, only this region. So this VIR D1, D2 complex excises the tDNA from the TI plasmid. Okay, so then what happens? This single stranded tDNA along with VIR D2 and VIR E2. Okay, so three things: tDNA, VIR D2 and VIR E2. Okay, so this complex of DNA nucleoprotein complex, this gets entry from the bacterial cell to the plant cell, and this is mediated by this protein VIRB. So VIRB actually helps in making the smooth smooth passage of this nucleoprotein complex from the Agrobacterium tumifaciens cell to the plant cell, and here one transmembrane protein also plays another role. Okay, transmembrane protein is also there, which also plays important role in this passage of the nucleoprotein from the plant and into the from the bacterial cell into the plant cell. Now what happens, as soon as the single stranded tDNA comes into the plant cell, it becomes covered by E2 VIR E2 protein. So this is the tDNA. Okay, this is the excised single stranded tDNA, and as you can see here, it is covered by whole lot of VIR E2 proteins. Okay, so these are the VIR E2, this small one, and this big one at the end, this is the VIR D2. Okay, so same nucleoprotein that entered from the bacterial cell, that came from the bacterial cell, is now inside the plant cell. So this nucleoprotein is actually made up of tDNA and two proteins, VIR D2 and VIR E2. And what happens, this nucleoprotein complex enters the nucleus of the plant cell through nuclear pore complexes. Okay, so nuclear pore complexes are small pores which allows the free passage of movement of molecules from the nucleus into the cytoplasm and vice versa. So these are mostly present in like plant cells and others, mammalian cells. So what happens, this nucleoprotein complex enters the nucleus from the cytoplasm. So this portion is the cytoplasm and this portion is the nucleus. Okay, now what happens after it enters the nucleus? So this tDNA is integrated. This tDNA is integrated inside the genome of the host plant. Okay, so inside the genome of the host plants through a mechanism called as the recombination. Okay, there are many sorts of recombination you can study in molecular biology, which are non-homologous recombination, homologous recombination, these things. So by this recombination mechanism, the tDNA gets integrated into the plant genome. So what is the biological significance of this? So biological significance is that if you can suppress the virulence, okay, if you can suppress the virulence, means that part, only that part of the virulence which has disease causing capacity. Okay, because otherwise what will happen, this will, in normal situation, what happens, this Agrobacterium tumifaciens causes a disease in plants, and that disease is called as the crown gall disease. Okay, so crown gall disease. So this is what happens during normal infection. So if you can suppress this disease causing ability of the Agrobacterium tumifaciens by any sort of modification, and in the tDNA, if you can insert along with the tDNA, because the tDNA has itself, with the help of certain proteins, it has its own integrating property because of the genome. So if you can insert your gene of interest, okay, with the tDNA, gene of interest with the tDNA, so what will happen, because the tDNA is getting integrated into the plant genome, okay, because of its specific sequence and function, so your gene of interest will also be inserted into the plant genome, right? So in this way, what happens, you can bring about your desired genetic change, okay, or your desired sort of mechanism into the plant genome. So you can insert your gene of interest, whatever gene you want to insert, through exploiting this natural capability of tDNA, of integration of tDNA. So you can exploit this step and make your own genetically engineered recombinant plant cell. Okay.